Yes.
In the calf. I take like
Okay,
a
so
handful.
let's go ahead and get started. I'm going to, of course, do the recording. There's a lot of material for this test, obviously. You guys are probably aware of that.
Scared.
So, I am going to try to hit the high points, but you guys need to ask questions if you have them, all right? Some of the stuff is going to be, I'm just going to flip through it because unless you have a question, you should be able to just get it from your notes, right? There's no big mystery about it. So, we'll start out with this. Any
Can
questions?
you go to the partial pressure
I have a
stuff?
question,
Yes.
but I need to get on there first.
Okay. We'll come. Just keep it in mind. We'll come back.
Can we
Okay.
go to like the examples of these?
Yes, that's probably a good place to start.
Yeah.
These other things are pretty normal,
important.
right?
Everything else is like pretty straightforward.
Okay. So let's talk a little bit about partial pressures. You should be able to calculate partial pressures. Can you do that? Right. So again, if we talk about, let's say the atmosphere of Pluto, the pressure is, well, be high, 10,000 millimeters of mercury, and let's say it's 30% methane gas, right? What? Man, that's why I'm gonna table this one. What would the partial pressure be?
Times that 5.3.
Yeah, so 3,000. Okay. Right? So again, it's just really that every single molecule of gas is contributing equal amounts of pressure. So if you know that 30% of the molecules of gas are, in this case, CH4, methane, you know that it's contributing 30% of the pressure, so you just do a multiplication. Right? That's the idea of partial pressure.
Mm-hmm.
Yes.
Now, are you going to give us the percent on the exam, or are we going to?
Oh, yeah.
Okay.
Yeah.
Okay.
So that's 3,000 CH4 in Pluto.
3,000 millimeters of mercury partial pressure for that. The reason this is important is because if we know partial pressures, we know the direction things move. Right? So things move from high to low partial pressures. So that's why it's important to know that. Okay, so let's think about some of the stuff that goes on in the alveoli. Right? Not to be confused with the areoli. That's what we were talking about earlier. All right. So here's the alveolus. And again, what we're concerned about is partial pressure for oxygen and partial pressure for CO2. And we could talk about that and the partial pressures that are in the blood.
Is this for quiet and inspiration and expiration?
Quiet, yeah. So again, some normal numbers, 104 and 40.
MMHG, right?
MMHGs for everything. I'll leave those off, that's the units. Okay, now if we think about what's going on, if I'm doing quiet inspiration, expiration, I'm breathing in about 500 mils. My lung volume, when it's not expanding a lot, is like, let's say, 3 liters. So I'm bringing in 500 mils, that's all equilibrating within that 3 liters, right? And so when I breathe out, I'm not breathing out exactly the same 500 mils, I'm breathing out a sample of what's in my lungs, right? So that means that, what I breathe out is really from 5 or 6 breaths ago, right? It's kind of the average of what I breathed over the last 5 or 6 breaths. So what that means is that there is less oxygen here than there is in the atmosphere. Because as that air is set around for a while, I've removed oxygen, right? Where did the oxygen go?
Would you?
It would, huh? Where the oxygen go? It went into the blood, right? CO2 levels have built up because CO2 from the body has bled into that area, okay? So let's imagine I start
how do
to...
you know it into the blood? What is it in the blood?
It's, every time you breathe in, there's oxygen that goes to the blood, right? So it goes to tissue. You also know that CO2 leaves the blood so it can go out into the alveolus,
Okay.
right? That's the whole external respiration thing.
I thought that was when we had the pressure differences and to know.
Well, we do. I mean, this is, for example, it comes in at, 60 and
140.
40? Or 45, I think, right? So there is this difference. So air does move, or I should say gases move.
So does oxygen always go high to low, like from in the thing to the blood, and then for CO2 it always goes blood to...
Yeah, it always goes high to low. That's just the way it's things move down a concentration gradient. They move down this partial pressure. So you think of it kind of like concentration gradient for gases.
So
right?
could the oxygen be like higher in the blood? and then it go the opposite way?
Well,
Or is it always
yes,
gonna
but
be-
you wouldn't typically do that because you don't want to do that.
Okay.
But yes, it could if you were to like.
But like this
Breathe
is like
pure
mostly
oxygen,
the
yeah.
normal way to
Yeah.
do it.
Okay, so I breathe in more deeply. Breathe out more deeply, right? So I run a 50-yard dash and I'm like huffing and puffing at the end of it. All this gas exchange is happening. Ventilation is occurring, okay, at a high level. So what happens to the level of oxygen in the alveolus? Right? Because we're bringing in more of the outside atmosphere in. So it's more like the outside atmosphere. It could potentially go to 160, right? Probably wouldn't,
Wait,
right?
so when you're heavily breathing, it goes up?
Mm-hmm. Because it's more like the outside atmosphere.
What's the outside atmosphere?
It's like 160 millimeters of mercury at sea level, right? And does this go up or down?
Yeah.
Down, right? The outside atmosphere doesn't really have much CO2, right? So it would go down. Okay, and I'm exaggerating. 'Cause the other way you could think about it is what happens if I hold my breath? What happens to the oxygen levels in here? They start going down, right? What happens to CO2 levels? They start going up. Right, and so basically, again, the what's in the alveolus is more like the body. If I bring in more air, what's in the alveolus is more like the outside
So
atmosphere.
then what would the blood be like? The same?
It's going to equilibrate with whatever's here, right, under normal circumstances. So this would be building up. Yeah, I mean, we talk about some methods, and we'll get when you talk about ventilation perfusion matching. You do try to make sure you always have a certain level of oxygen and CO2. Right, so these numbers that I give, 104, 40, 60, 45, are kind of standard, right? And you're going to try to maintain that. Okay, does that kind of make sense to people? Hopefully.
So on the test, will you give values and we just have to know which way it's moving?
Yeah, I mean, I might ask you to indicate if things are going up or down in partial pressures.
Yeah.
Or make up a value that's realistic, right? And what I'm looking for, does it go up, right, or does it go down? I don't expect you to memorize these values. Okay, so here's our, partial pressures for oxygen. Actually, this is 40. That can't be right, is it? Whatever. It doesn't matter that much. Right? I told you numbers don't matter, so it doesn't. Okay. Okay, so hopefully that's pretty good. You guys pretty good with hemoglobin?
Yeah.
Yes, hemoglobin carries which gases?
O2 and CO2.
And CO2, right? Which one is going to be on the protein portion?
CO2.
CO2, right? And the heme group has the oxygen.
Yeah.
You guys can do partial and fully saturated percent saturations,
Yes.
right? I won't ask you to do any complicated math with that.
What?
You should be able to, kind of have some ideas. All right, carbon dioxide transport. I could go over this, but I think, we'll get there in a second. But this is something that's covered pretty well in the book. Do you think you guys can handle this?
Yeah. The bicarb.
So is this going to be like a
Actually,
diagram question on the test?
Vaguely remember.
I won't. This is a little much.
Yeah.
But I might ask you, like, how is most CO2 carried back to the blood? And you should know it's bicarbonate, right? But there's no sense in like having you do.
Like the chloride
gigantic.
shift and the
Yeah, it
carbonic
might be a little
and
much. There's
anhydrous.
plenty of other stuff to ask about on this test.
So what do you want us to remember, you said?
That the bicarbonate is the major way you carry stuff back.
Back to what?
Back to the blood in general, right?
Carry anything or just CO2?
Just CO2. Yeah, I don't know. I guess it's that's a lot. Right. Minute ventilation.
The amount of air a patient moves in 1 minute.
Correct.
Bingo.
Yes. Right.
Wow.
It's like cardiac output for your lungs, right? And again, it ranges from 5 liters to somebody who's really highly trained athlete up to 200 liters per minute.
Okay.
So I think you're going to miss us next semester.
Oh yeah. I don't know how I'll handle it. I'll see some of you guys around, right?
Yeah, I'll be in this building like
Yeah.
24/7.
Crying our eyes out.
Yeah, seriously
Sobbing
crying.
my way through nursing if we make it there.
All right. Controlling breathing. Right. Which one of these is the most important?
CO2.
Okay. How do we measure CO2 levels?
The carotid receptors? No?
It's
No,
it's
cerebral spinal.
Cerebral spinal fluid. Okay. We measure oxygen levels with the carotid receptors, but we measure CO2 with cerebral spinal fluid.
Why is the CO2 the most important?
Yeah, it's because it only can vary by a small amount. Right. So here, for example, it starts at 40 millimeters of mercury. If it goes up or down 7.5% or 3 millimeters of mercury, you start changing your breathing. Whereas with the oxygen, it has to go up or down to like 40%.
And then O2 is measured by the carotid receptors.
Mm-hmm. Right. I guess technically you guys should probably be able to know something about how we measure stuff in the CSF. And again, remember the whole idea is because there are no buffers, the oxygen, or I should say the CO2 that goes into cerebral spinal fluid changes the pH a lot. And we're essentially measuring pH to determine what's going on with CO2.
It decreases the pH, right?
If there's more CO2, it would make the pH go more acidic. If there's less CO2 than normal, it would make it go more basic.
Okay.
So there's no buffers, you said?
Yeah. I guess we didn't do ventilation perfusion matching.
Oh,
we did,
man.
but you didn't put it in.
I didn't pin the gray sets. That's a real shame.
Oh,
Dude, thank God.
I think I'm getting enough in the greatest.
Yeah.
Yeah, we will-
Okay.
I mean, you put 117 slides up in here.
That's a lot of slides. I agree. Okay.
Yeah, usually it's like 80, 90. And
Okay.
Usually it's like 75. Yeah.
now we added another 30 on it.
All right. So the anatomy of all the stuff we talked about, all the different cells we talked about, you guys should be able to kind of do that. But let's kind of think about this a slightly different way. One thing you might do is think about, a meal that travels through your digestive system. Okay? So let's have an esophagus, right? We've got the stomach, we've got the small intestine, right? We've got the large intestine, right? And there's our anus, okay? And then of course, what else do we have? What else do we have from the small intestine?
Kidneys.
Not kidneys.
Wait, not kidneys.
The
Okay, that was
pancreas.
Pancreas,
Liver,
Gallbladder.
gallbladder.
liver, and gallbladder. I'm just going to draw the gallbladder because the liver and gallbladder are essentially doing the same thing for our purposes, right?
Mm-hmm.
They're making what?
Bile.
Bile.
Okay.
Can we label what is what on that?
Yeah, it's a little bit weird. Okay.
I'm not even gonna try. I was trying to
Stomach.
do that, but
Okay,
I
okay.
don't know.
Small intestine, large intestine, anus, right? Esophagus.
Oh, so it's drawn sideways.
Right. Yeah.
It
I'm
seriously
not-
looks like the
I don't know about
Some
you.
lips here.
and the,
Yeah, that we did
the
today.
afferent arterioles. Those look like the arterioles.
Hey, that's on the next exam. I don't want to think about that.
All right.
Hey, man.
So let's think about. Let's think about if I just drink water, and let's say I drink a soda or pop, as I would call it in Kansas. Okay.
What about
I'm from
you?
Kansas, you guys know.
Oh,
Yeah. So what happens? Where do we absorb stuff? Right. Let's just say we drink some pop. Do we absorb anything in the stomach?
No.
No. Okay. What about the small intestine?
Yes.
Okay. How are we going to break down? Well, actually, we don't have to break down anything to the simple sugars, right? We don't really have to break much down, right? We just basically, let's assume that we're getting like one of those Mexican Coca-Colas that has real sucrose. We have to break down the sucrose into glucose and fructose. Where do we do that?
The small intestine.
Correct, but where in the small intestine?
The duodenum.
In the duodenum,
Duodenum,
yes. Do we need enzymes that are in the lumen?
Yes. No.
No, right? We have enterocytes there that have cell surface, enzymes that do final digestion and absorption, right? Ultimately, where does that material go? The carbohydrates that we absorb?
Say what?
Those carbohydrates, where do they end up going?
Can we go back into the blood?
They go in the blood, they go to the liver. Okay? So that's fairly simple.
'Cause
Now we'll...
Everything is going to the blood supply in the liver, essentially, right?
Yeah. So sugary drink is the first thing. So the second thing, let's talk about... Are you one of Dr. O'Shaughnessy's favorite things? Potatoes. Okay. Which is apparently true. I thought that was just a rumor that people Irish eat a lot of potatoes, and no, he does make a lot of potatoes. So potato, okay? I chew it up, goes to the stomach. What kind of digestion occurs here?
Mechanical.
Mechanical, right? Because there's no digesting the carbohydrates, but we're still going to mechanically digest it.
How are you mechanically digesting it again? Because I thought peristalsis.
Yeah, I thought
Not peristalsis.
Oh.
What does the stomach do?
Churning.
Churning, yeah. So it churns it. Still has to churn to break it apart.
What's that called?
Chime.
The-
No, the
the-
churning.
Just churning.
No, there's something didn't you have something like kneading like
Segmentation is different. Segmentation is
Oh.
small intestine. So churning is a special muscle movement of the stomach. How can it do the special muscle movement?
Distension?
What allows it to churn?
Muscular external.
Yes, but why doesn't every other place churn? Why does only the stomach churn?
Isn't it because the layer of, it's the third layer, right?
Yes, it has a third layer, right? It has an oblique layer.
Yes, girl. How'd you know?
All right, so we're going to mechanically digest it. It's going to arrive in the small intestine. What do we call that stuff? Chyme. Okay, so it arrives. How are we going to break that down?
You use like amylase to break it into disaccharides, and then.
Okay, where are we going to get all those enzymes?
Those come
From the pancreas.
From the pancreas. How are we going to liberate? Make sure those enzymes come out. What's the hormone?
pancreatin.
Yeah.
CCK.
CCK. Okay.
CCK.
Okay, and it goes through, again, the enzymes break it down, right? Final digestion is done by the enterocytes, and we absorb all that material, okay? Let's say I drink a cup of olive oil,
Ew.
okay? Stomach, no mechanical digestion, all right? I start to get in the small intestine. The small intestine is going to like that.
It's not gonna like it.
Not really.
You're drinking straight oil.
Oh yeah.
You drink oil, like what did we say?
Lipids.
So when we talked about different phases, right? And so part of the intestinal phase is to be concerned about lipids.
Yeah.
And so we would release what to stop this?
Bile.
Don't even be gastrin.
Gastrin? Well, no, gastrin causes the stomach to be more active.
Secretin.
Secretin and CCK, which are both gastrin inhibitors, right? And so
That's going to slow it all down.
slow it all down. So the secretin, the CCK would cause us to release bile, right? And some pancreatic enzymes. There are some lipases from the pancreas. They're both going to go here. And we're going to slowly break down those fats. They're going to be absorbed again by, into the micelles and then the chylomicrons. And then we're going to transport it into a specialized structure that's found within the villi. What was that specialized structure called?
Ooh, it's in the center. Lacteal.
Lacteals, okay? The juice that's there is called chyme. Chyle. And then we're going to absorb that back. And finally, that's the juice and the lacteals. Let's just have a totally meat thing, right? Eat a pound of chicken, not much fat, all right? So let's go to the stomach.
Where are the lacteals?
They're in the villi,
Center of the
right? Those little things that look typically
In the
big.
stomach.
It's like that space.
And small
area.
intestine.
Oh yeah.
So then I eat just meat, okay? So what happens in the stomach?
Breaks you down even further.
Churning, but there's going to be some other digestion that happens here, right? Is there going to be chemical digestion of proteins? Okay, so how do I do that? Gastric juices. So this is one of the things we talk about the gastric phase, that if there's a lot of protein, gastrin levels will go up. You'd make more gastric juice, right? You would also make more of an enzyme that breaks down part proteins. What's that enzyme called? What's the enzyme that breaks down proteins called? It's a little trick question because we're going to make one enzyme and then it turns into something else.
Thymogens.
Pepsinogen, which converts to
Pepsin.
pepsin. Okay? So we're going to break things down. Again, we're going to have some chyme enter. Again, if we have high levels of proteins, we're going to release CCK, right? If this would be an acidic environment, so the stomach acid, is going to be levels are going to be high. So we're going to enter the small intestine. So in order to counter that juice, what do? We release what?
Is that when you?
Can you repeat that again?
So in order to counter the fact that we've got this acidic juice coming into the small intestine, what do we do?
Inhibitors, the gastrin
Yeah.
inhibitors.
Well, we do release gastrin inhibitors, but we're also going to release something else that has a high pH that counters stomach acid.
Is that the
Bile salts. No.
Has a lot of bicarbonate in it. It's made by duct cells.
Renin?
Hmm?
Renin? Whatever?
No, that's for
No, that's very.
It's we're talking about pancreatic
stem
juice.
cells?
Pancreatic juice, right?
wait, hold on.
Oh.
Right. So remember, the pancreas makes two different things. It makes all these enzymes that are under control of CCK. It also makes pancreatic juice that are under control of secrete. All right. So that kind of covers it, I think. If you ate a lot of fiber, your stomach would expand a lot, so you'd have some gastrin, but there's not much to break down. Things would move pretty slowly through your small intestine, right? But eventually you would lose all the nutrients, so let's just say I ate a lot of broccoli, okay? There's not much nutrient value, so we basically don't detect much nutrient value. Very soon we don't detect much. Motilin levels build up. What happens when motilin levels build up? Not the large intestine. Small intestine contracts. Remember that idea of those waves that start and then move a little further and start and move? Okay. If we're in the large intestine, we can actually digest some of that fiber. What digests fiber in the large intestine? Yes. Yeah, the flora. Okay. And remember the rabbit that has to eat his poop, right? It's digesting stuff in the large intestine, and then it poops it out and eats it. Or just be lucky you're not a rabbit. Right? I just feel good you're not a rabbit. Okay, so that's a lot of putting a lot of things together, and I definitely would, I might ask you some subset of that on the test, but that's the kind of thing you should be kind of doing to think about things, to see if you understand stuff. So follow some food through and see what would happen. How would it be treated? Okay, so I'm just going to go through. And again, a lot of these are just kind of basic facts. I'm going to be surprised if you saw this on the test or some element of it. The layers. This organ, incidentally, is what organ? What organ is that?
Is that the small intestine?
Small intestine. That's kind of our norm. We consider that the normal organ, right? And everything else is kind of variations from that. Aaron, did you have a question?
I have a question about CCK.
Mm-hmm.
What happens when it says like CCK triggers bile release?
Yeah.
Is it high amount of protein and high amount of fat so that it then releases bile? Like, because in your notes it just
There's
says,
several different things that triggers CCK release because it does more than one thing.
Okay.
So, high levels of fat trigger it.
So high level.
Yeah, high levels of protein trigger it because it also is used to get these enzymes out. In reality, what's going on is that depending on what you have, you're going to have different levels of CCK release, right? So in other words, if I just eat chicken, I'm going to have a certain amount of CCK release based on the fact there's protein there,
And what about
right? If I eat something like a fatty piece of sausage or something, I'm going to have more CCK because there's fats and proteins there. And there's going to be slightly different responses to those different levels.
and low pH triggers it too?
Yes, that actually triggers more of the secretin because we want to counter the pH by releasing pancreatic juice. So in general with the hormones, once you kind of learn them, you can kind of guess what they do because you kind of think about what they're trying to accomplish, right? Okay, so let's see. I think. All right. So mostly good so far, right? Critical picture. That's all I'll say.
What is peristalsis again? That's when you that's like squeezing of the tube.
Mm-hmm.
Like
Yeah, so
toothpaste?
it's above the food it's pushing and contracting, below it it's relaxing, and there's a wave that pushes things along.
So it's contracting above the food and
Correct.
relaxing.
The esophagus is the best example of peristalsis because that's all it does, right? Segmentation is kind of that kneading thing. We watched that movie where the person's small intestine was doing like this, right? Yeah, so that's segmentation. And churning is that thing where I showed the pig stomach and you had that complicated motion. Yes? Question? Yeah.
No.
Oh, I'm administrator. Okay. Okay.
What was segmentation?
When you have the kind of a kneading type pattern, and I showed that movie, The Operation. The small intestine was kind of moving around a little bit. Okay.
Are there any specific functions of the mesenteries that we need to know?
Yeah. Sort of. Again, this is the stomach, and you guys can't see it in the recording. We've got a picture of the stomach, right? And there's an outer curvature, right? And off of that hangs the greater omentum, which is a ventral mesentery. That ventral mesentery is mostly about fat storage. There are some immune cells there as well to deal with peritoneal infections.
Where is this one?
That's hanging off the greater curvature, right? And then if we were to look at the stomach, we'd also see there's a lesser curvature. That's a lesser omentum. That's an example of a dorsal mesentery. They're blood vessels that are coming off the stomach that are carrying that blood back to the liver and the circulation in general. So when you think about the dorsal mesenteries, they're anchoring organs in place. They're allowing blood supply to go to and from the organ. There's nerves that are coming to and from the organ through that mesentery.
Okay.
All right. So again, you guys need to know about parietal cells and sheath cells. The idea that acids denature proteins, right? So again, if you put an egg in vinegar, it will cook. Right? Gastric juice, gastrin and gastrin inhibitors. We talked a little bit about that, right? Remember, most of the gastrin inhibitors are also playing other roles. So CCK and secretin are kind of the two classic gastrin inhibitors. Right? Why do we have the bicarbonate in the mucus here? Yeah, what's the use of that bicarbonate there? So we make mucus and got by some goblet cells.
Helps aid things to go through, right?
Well, the mucus is of course making things slide along, but this is actually the stomach, so we're not so worried about that. What are we using the mucus for? Why does the mucus have bicarbonate in it?
Oh, for more of the acidic pH environment, right?
Yeah, so if the protons start to go in towards the tissue, they interact with the bicarbonate. So again, this is the idea of a buffer. Remember, buffers are systems that resist changes of pH.
So bicarbonate is the buffer
Mm-hmm.
to keep the pH
From basically trying to keep it neutral, if possible, in the mucus, and then down onto the tissue. Okay. All right. Are you guys mostly okay with this? Again, we're flipping through. So the different phases we talked about. the cephalic phase. Again, we're going to make a little bit of gastrin. This is controlled by you thinking about food or seeing food. Okay. The gastric phase is controlled by what organ?
Stomach?
Yeah, stomach, right? It's named after the fact that it's controlled by the stomach. And so again, if we have a big meal and it causes our stomach to expand, we release gastrin. If there's proteins present, we release gastrin.
And HCl.
Yes, and then gastrin would cause gastric juices to go up. Right. Essentially, what happens is we release gastrin. That causes the parietal cells to make gastric juice. Right, and the pH goes down. So really, low pH, gastric juice production, and gastrin are all sort of tied to each other.
So this is part of the gastric phase?
Yes.
But I thought
Yeah.
it says it increases with pH, not decreases.
Well, I'll get to that in a second. Yes, Aaron?
What is a G cell?
That's the cell that makes gastrin.
That's
Yeah,
what it is?
that's the cell that makes the gastrin. So it's an endocrine cell. Okay, so Elizabeth asked, why does it say increase pH? So let's go back to this idea of proteins are cooked by acids. If I throw a protein in acid, what's going to happen is some of those protons are going to start interacting with the protein. And the pH of the liquid would go up a little bit because the protein's sopping up some of the protons. And so one of the things that tells the stomach that it needs to start making more acid is an increase in pH, which suggests there's something absorbing pH, which is probably food absorbing it. And so if something is absorbing acid, so you probably should make some more. So that's what that means. The plus means we're going to make more gastrin because there is an increase in pH.
So proteins increase pH, and because the proteins are increasing the pH, it releases gastrin to make the pH go back down.
Correct.
Mm-hmm.
Yeah. So the pH suddenly going up. So this is the idea: is the stomach's sitting there, also the pH goes up. Well, why'd that happen? Well, it's because proteins are present, basically. Right. So it's another way to measure the protein levels. All right, intestinal phase, again, the idea here is the small intestine is also monitoring things. It's monitoring what's going on in the chyme. And so it doesn't like acids, right? It doesn't like proteins. It doesn't like fats. It would like to digest those things very slowly. So it would release gastrin inhibitors to help slow down the stomach whenever there's too much acid, too much protein, too much fats. Yeah?
So when the small intestine is in distension, the pyloric sphincter closes?
Yeah, that's the other thing that could happen. If there's something really big that comes through, like let's say you don't listen to your mom and you swallow something, you swallow chicken bone, right? Chew properly. The pyloric sphincter is not going to like that. And so it will close down. It won't let it through. And then when you're asleep, it will open up and let it through. Right? All this stuff happens when you're sleeping. Bigger items do eventually pass when you're sleeping, but during normal digestion they don't. Yeah.
So when there's too much acid, protein, and fats, gastrin inhibitors are released. Is secretin released as well?
The secretin is a gastrin inhibitor. Yeah.
Is it? Okay.
Yeah. So again, there's a lot of these things are playing dual roles. Yeah. So that does two things. It slows down the stomach. It also enables us to start making pancreatic juices that buffer out the acids. CCK is also released. That slows down the stomach, but also brings in things that help us digest fats and enzymes that help us digest proteins. Right? So it all kind of works together. Okay, we talked about the fact that, yeah, big things don't go through because the pyloric sphincter closes. All right. There's the churning model. Yes?
What does the jejunum and the ileum do?
There is some stuff that's only going to be processed in the jejunum and ileum. So we're really focusing on the duodenum, but there are things that are specifically absorbed in those areas. The ileum has some specialized features that had some more amino tissue, right? The Peyer's patches that we mentioned. But for our
Immune
purposes, huh?
Immune tissue?
Yeah.
Okay.
Right, because it has to worry about weird stuff coming from
A large
large intestine.
And
Right?
the duodenum does all the absorption basically. Like
Pretty
you said.
much, for our purposes, yes.
Okay.
It's a major absorption. And think about, one of the things that's happening in the duodenum is it's actually putting a lot of fluid out there. Right? There's all this pancreatic juice that's arriving. There's all this gastric juice that's arriving. There's saliva that's arriving. As you start to absorb things, it turns out that you also put liquid just from the sides of the small intestine into this pot. And so the other thing that happens to the jejunum and ileum is we start pulling some of that water back. So we're absorbing water also in the jejunum and ileum. So once we've got all the stuff kind of digested, we also bring back water into your body. And so by the time you get to the large intestine, you've removed a lot of the water. You also do that, of course, in the large intestine too. All right. So again, this is all the small stuff.
Wait, so the villi in the small intestine, what does the capillary bed do with it?
So at the center of each villi, or villus actually is plural, there's a capillary bed, right? And remember the cells that are on the surface of the villi are doing all this absorbing. And that material goes into the blood supply.
So, but I thought the lacteals were in the middle of the villi.
They are too, right? They're right next to them. They're easier to see, so typically you often see just the lacteal, but they're both there.
Okay.
So this is absorbing fats. This is absorbing carbohydrates, nucleic acids, vitamins, kind of everything else.
That's what the lacteal cells
Mhm.
absorb.
Yeah, they both go back eventually to liver.
Wait, so lacteals absorb fat,
Yes.
and the capillary beds absorb basically everything else.
Everything else. Okay, so here's where we kind of get to making some of the enzymes and things like that, the ducts and acinar cells.
The enzymes aren't active until they reach the small intestine, right?
Correct. Right, what would happen if they became active in the pancreas?
They start digesting the pancreas.
And that can't happen. There's pancreatitis can occur where those enzymes become active early and then they start eating up the pancreas. So that's a really problem.
What does Imogens do?
They break down proteins. I won't expect you guys to memorize that. Right? I do want you to know pepsinogen and pepsin, but the zymogens are just a whole class of things that break down proteins, and there are several of them around. Why do we need bile?
To digest fats. and
Yeah.
stuff.
So it takes fats and makes them into little teeny balls, right? That we call micelles, and then those are brought into the cell. They can just go through the cell membrane. They're repackaged in these things called chylomicrons, right? And then they're going to the lactose. Well, there are a lot of slides here.
Yeah.
Oh my goodness, there's a lot of slides. Okay,
Different.
so this is another picture that could be on the test, right? Just saying.
Can it just be all
Labeling.
Yeah.
Well, that's partially what the lab is too. Okay, so let's. Oh my God, there's a lot of slides. All right, let's do immunity. This is where it gets interesting, right? Okay. Okay, so surely people have some questions about this.
Yes.
Yes.
Is there a way to is there a list that we can make between the difference between innate and adaptive?
Sure.
Yeah, I don't understand how you can tell the difference.
Yeah, that's what I'm.
Okay, so let's do it.
Okay, do it.
I'm in.
Did you guys see the thing about South Dakota's new anti-meth thing?
What?
Huh?
they got this new thing about anti-meth, but the slogan is meth. We're on it. and it's pretty funny, but it's a little crazy. Okay, Google it when you get back home. All right, so let's talk. What is the primary process associated with innate immunity?
Wasn't that the
Inflammation. Okay. Now, I'm just going to do some of this on the board, and then we can go into some details. What's one of the major cells that's involved in inflammation? The cells that actually eat bacteria, for example.
Oh, the macrophages.
Macrophages. Okay.
I did all this.
So here they eat bacteria and release cytokines.
Which are like, that's when there's the cells of movement. So like they move to the specific area.
Yep. So the neutrophils come. Are macrophages only part of the innate system?
No.
No. They are part of both. Why? What else does a macrophage do?
It can make antibodies or antigens or something. No.
Can present antigens.
Present antigens.
Okay. If I have an antibody on my surface, I'm also more likely to be eaten by a macrophage. So IgGs help identify things for macrophages to eat.
What is it? Macrophage? What?
If you have an IgG on you,
What is that?
What's
that's
the
an
antigen
antibody.
or antibody.
But what is the thing between IgG and macrophage?
An enzyme?
And.
Yeah.
Okay, antibodies are kind of a classic thing that are associated with the adaptive system. What makes them? Plasma, B cells, okay?
B cells.
But you can see that I can use that antibody both ways. If I have an antibody, I also can activate complement. To produce the MAC, membrane attack complex. But there's also ways to activate complement
That's the C3, right?
to MAC that are purely innate. Let's see, what else do we have?
So like, how can you explain that they are both? Like, are you gonna have like short answers where you have to be like explain if this is?
I would probably say explain if they're both.
How do you explain? Like, you just.
You would just say, here's a macrophage. Tell me, and you'd say it's both. Well, give me a role that it plays in the innate immunity. Give me a role that it plays in adaptive immunity.
Okay.
Right? Yeah.
I do have.
So another one, CD4.
So the macrophages produce antibodies?
No, they just, if you have an antibody on your surface, a macrophage is more likely to eat you. So it increases macrophage activity to have antibodies on something. So, well, I guess it could, CD4 is both, right? It's part of the adaptive, but it also coordinates part of the innate system. So yeah, I mean, there's a lot of things. The big ones are macrophages and complement.
What do
Right.
you mean the macrophage complement? Like I'm confused. Like the IgG plus complement to the MAC. What do you mean? Like what?
Okay, so let's. Okay, so this is all, yeah. Okay, this is probably more profitable once you've done a little bit of study. Okay, but did you guys? If you want to take a little picture, I don't know. Anyhow.
To be honest, I don't even remember going to some lecture. I don't recall IgGs
I don't even.
ever.
IgG?
The
I
IgG.
don't
I don't think we did that. I don't think we did that.
See, I don't know because I don't recall it. That's why I don't recall it.
Oh, okay. So let's talk about a few
I remember
things.
the map.
All right. So we've got complement. Okay. So essentially, what's going to happen, and this is this is kind of how we talk about it. It is an oversimplification. Is that you have a cell. It has, again, certain antigen is in it. Okay, you guys should be able to find an antigen. Some of these are very commonly found on all bacteria, for example. And that is going to activate some pre-existing proteins to bind to that. That is going to attract those complement proteins to the area. And we're going to make the MAC. Okay? Again, this is really a small subset of things that we're looking for. Something like 15 to 20 different proteins that look for common themes in bacteria, attack that bacteria. If we have an antibody, right? No, this is a different color so we can see this better. Which were made, again, this is part of the adaptive response. If we have an antibody, that also attracts the complement and places it forward, right? But this is really specific. We can make something to a very specific thing.
Mm-hmm.
So it takes longer, but this is actually a little bit more powerful because we can identify more things on this cell. Right? We give the example of a bacteria from Mars. There's no pre-existing way to attack that bacteria. There's no way to identify that. It's alien. We could actually make an antibody to it, though, and then we could activate the complement. Right? So that, again, and we talk about various types of antibodies. The most common type is the IgG. It's the one that looks a little bit like a Y.
Mm-hmm.
Correct?
Yeah, that's where it has like the
Oh, I remember that.
different variables
Right.
and stuff that.
So it has the variable regions and the constant
Yeah.
region,
Uh-huh.
right? Again, it's the most common type. There are other types that really just have different constant regions, but it can have the same variable region. Right? How many different types of antibodies can we make? A lot, right? And we talked about that sort of system that we use, the VDJ
Yes.
system,
Mm-hmm.
the somatic
With the different
recombination.
combinations.
Yeah. Do you guys understand the VDJ and somatic recombination?
Is that going to be asked? Like, are you going to ask a question about that?
I would probably ask you to at least define it.
Is that
Potentially.
like greatest hits?
what?
Yeah.
Is it?
That's my only.
I don't
That's
know.
not
Oh, man.
I
That's
don't know.
the
It
coolest
was
biology
the
we talked about.
title with the.
Yes.
Oh, looks like you can't include
A bunch
it.
of
Oh,
Yeah.
oh snap! There
Right
it is.
is.
Okay.
Y'all really had to say
So like, what is it?
So what is it? Okay, now everybody cares. All right. So the idea here is that we have to do two things. We have to generate a lot of different types of antibodies, right? And we have to make sure that once we generate the antibody, that the cell only ever makes that type of antibody, and it only has that particular type of receptor. So what we're going to do is we're going to basically make a random sequence to build a receptor and an antibody, right? So here's, for example, a B cell. It has a receptor, right? There's a constant portion to it. There's a variable portion to it. The IgG that it makes has the same variable portion, right? So it's going to recognize the same thing. We want to generate a lot of different variable regions, okay? So the way we do that is there are several different genes that are spread across the genome that we use to build the receptor. Okay, and so on this image, they're showing you essentially what you're going to do, and I think I gave the Lego model for this in some of the classes, but I
Yeah,
also
you
did the
did.
Chinese buffet. So one thing you can imagine, you go to a Chinese buffet, and there's all these meat dishes, right? There's 40 different meat dishes. You pick one at random. And then there's several different vegetable dishes, which you pick at random. And then there's some desserts that you pick at random. And you're going to build a meal from that, right? The same thing is going on here. We're building a gene, right, and some mRNA by selecting different portions. And so we do that randomly so we can build a lot of possibilities. Again, you don't have to pick just one. You can have 3 meats, right? Or you can have 1. So what you do is you have this possibility to build a lot of different types of meals from that. So that's where we get the variability, that we're picking different gene sequences, putting them together in a messenger RNA, and using that to build our protein. Now, where it gets interesting is the somatic recombination part, because if we go back to the buffet, what we find is all the dishes that we did not pick the first time have been removed. So all the gene sequences that are picked to make the receptor the first time, everything else is cut out of the genome. It's gone from the DNA. And so now the only type of receptor that that particular cell can make, and all its progeny, all the daughter cells that come from that cell, the only protein that it can make is the protein that it made the first time. So that gives us variability, but it also gives us stability for that one particular cell.
How does it give us variability if we can only make one thing?
Because the next cell is making its own random choice. So you're making...
Like,
per
basically
cell.
you start off with like a bunch of different things, and
Mhm.
like a bunch of different things come in and pick one from each,
Correct.
and then they make different things.
Correct.
So
We're
only
literally
make
talking about you have billions of type of B cells, each which are doing their own thing. And when they get activated, then all their children are going to make that one thing, right? That's the idea of the clonal selection that we talked about,
Mhm.
where you have a naive receptor and it gets chosen and it starts to activate and make more copies. All those copies are always going to make that same receptor and the same antibody.
So it makes receptors and antibodies?
Yeah, the same cell is gonna the same variable region is involved with the receptor and the antibodies, right? Okay. So that brings us to the idea we have all these B cells that we produced. They have to be self-tolerant, which means what? They know not to attack self. If they attack self, they essentially will be killed. That's how they learn things, right? So basically you just kill everything that attacks self, and what's left over should not attack self, okay? They also are all immunocompetent, which means what?
That's when they can recognize one antigen.
Yes, they only recognize one antigen. Okay? So, again, that's sort of the basic things. And same thing is this similar process is happening in T cells too, to develop their own receptors, yeah.
So when you talked about the colonial selection,
Mhm.
that's when it has the two responses where like it's the primary and secondary, right?
Yes, so let's imagine, so this is a good point because this is something that will definitely come up on the test. Right? We have some cells that each have their own receptor. These are B cells, but a similar process could occur in the T cells as well. They each have their own receptor, and I'm going to select one. Right? The proper antigen finds its receptor. That's clonal selection. We've selected that clone. And that clone is then going to start dividing.
Mm-hmm.
Okay? And if these are B cells, what cells do we make?
You're making the antibodies or the plasma cells. I'm
Plasma
sorry.
cells.
So wait, what is clonal selection? I don't get it.
So you've got all these clones, all these different cells that are recognizing different things, and I've chosen one to divide because it's recognized as antigen.
Functional cells or something?
But if they all have the same antigen, couldn't they pick the?
Well, they don't all. They all have different ones. Right? That might be the only cell in the body that recognizes
The
that antigen.
antigens are binding to the specific
Yeah.
receptor.
It's binding to that. Remember, we generated all these specific receptors. So we have all these different specific receptor types, right? And so one of them is activated by that particular antigen. So we make plasma cells, and we make what?
The memory B cells.
Memory B cells.
But then isn't the memory B cell dividing?
It does make more than one, right? There's just less of them. So there's a lot of plasma cells, but fewer memory B cells. Okay? The plasma cells are going to make antibodies. They're going to live for a relatively short time, and they're going to make antibodies. And so what we see is the levels of antibodies would go up and then fall back down. That's the first time we've seen this, so this is the primary response.
Mm-hmm.
It's 5 years later, we see the same antigen. Secondary response. There are more memory B cells around. So we're actually going to we're going to be able to detect this more quickly because there's more of these around. They're spread throughout the body. And so what's going to happen is one of those cells is going to be, it's actually multiple of them are going to be activated. They're also going to make plasma cells, right, and more memory B cells. And these guys are going to make antibodies like before, but they're going to make more
Way
antibodies,
more.
right, collectively. And these plasma cells live for a longer period of time, so the levels of antibodies are going to stay higher for longer.
Mm-hmm.
Every time we activate this system, it gets more efficient.
Mm-hmm.
Right? So this is why now you are considerably less likely to get sick than you were when you were 5. You guys have experienced a lot of common things, and you have all these pre-made plasma memory B cells running around. You also have pre-made memory T cells running around. Similar idea happening. Okay, so, huh? of primary and secondary response. Right? And there's a nice picture of this in the book. It's a classic thing to ask on the test too, because it should be fairly easy for you to draw this.
So
It means fairly easy for you to grade.
to get from this, basically, as we get older, our immune system is a lot stronger.
It should be until eventually it does start to get weaker as you get older. It starts to kind of break down a little bit. Yeah, there's some point where it's at its height, right?
Yeah.
You've experienced enough stuff, but yet you still haven't. It's not weak either. So, okay. So we've got more time. We're not going to get done in an hour. Let's talk about T cells. This is everybody's favorite. APCs.
Yeah, my favorite.
So I thought that the APCs were like also with B cells too.
B cells are a type of APC.
Okay.
We didn't really get into it too much, but they are technically an APC. Okay, so we got two things to cover. We've got inflammation and we've got T cells. All right, so let's do some ideas with T cells. So the key thing here is first, we have to talk about cells in the body. We have to talk about antigen-presenting cells, which the macrophage is a kind of a classic version of this. And then we have to talk about CD4 cells and CD8 cells. Let me make sure I get this right, because I always screw this up sometimes. Okay. All right, so first, this is a cell that, let's say, has some sort of virus in it. Now, the way that we're going to be able to tell it has a virus in it is through the fact that it has an MHC1 that has something foreign on its surface. How did that get there? You should be able to describe how that got there.
There's many different ways you can get there, right?
Well, not for a normal cell. We're talking about a normal cell. This is a normal body cell.
Didn't it enter in then was coded?
Goes to the
No, that's
endoplasmic
APCs.
reticulum.
This is much simpler.
It goes to the endoplasmic.
Yeah, so we have proteins that are constantly being degraded. Some of them are part of the cell, some of them are not. Okay? They go to the ER, and from there they're put on the MHC1. Okay? So again, typically if cells are not any sort of problem, if they're not cancerous or they're not viral, what you'll see is a whole bunch of these little lollipop things that are just showing self. But here, because there's something abnormal, we're showing non-self. Okay? So the first thing with the MHC1s are kind of helping us to identify what's inside a cell. All right, this is for most cells of the body. Let's go to the APCs. They have MHC1s, and they have MHC2s. Right? This MHC1 is actually going to display all kinds of different stuff. Stuff from inside the cell, but also stuff from the environment, right? So for this guy, for example, it can get stuff onto the MHC1 through phagocytosis.
That's eating large molecules.
Yep, and breaking them down and then taking the amino acids and putting them on there. You can also get it through gap junctions, right? So it's randomly sampling what's in other cells because it forms a gap junction, and then what's in that cell flows into the macrophage, right? And then it closes the gap junction. And then it interacts with another cell, right? What's ever in it flows into it, right? Goes another one. And it's constantly doing that, making these random gap junctions. So what's in those cells, these little bits of protein are floating into it, and it can display stuff that it's observed on the MHC1.
So, in all of these ways to make it, they all end up going to the ER, right? Because they go to the ER into a vesicle
Yeah.
and out to the surface.
Yeah, because that's where we're
It's
packaging
just,
it.
It starts with phagocytosis and gap junctions instead of just being.
Yeah.
So with the regular way, is it just already in the cell?
Yeah, so it's
Like
also
for
doing the
not
same.
The APC. For
Correct.
normal
It's doing the
cell.
same process. Yes, this is already in the cell. Okay. the benefit of doing it these two different ways are that here, if it finds like a viral particle that's out there or a bacteria, it can find it. Okay.
From the environment.
From the environment. So it can be sort of like not even human, right? The gap junction thing is looking for stuff that's specifically in cells, and it can identify stuff that's in cells that are just like slowly developing a virus, right? And so it encounters stuff that is kind of hidden. And you don't want it to actually get to the point where maybe it breaks apart, and so you don't have to. The cell doesn't have to break apart for it to find it. Okay, and then the twos also can display foreign stuff.
But they're telling it not to destroy that.
We'll get there in a second. Right. Phagocytosis, it's equivalent to phagocytosis, slightly different, but that's what goes on in the twos. Okay? So, what we're going to do is that these MHC1s that have something foreign, and I have the green for the green light for destroy, right, are going to activate CD8s. So if these two cells come together, and this has a proper receptor for that antigen, this would be activated.
So the MHC1s activate CD8.
Correct. Right. And this one is going to activate CD4, right? And there's a nice little slide that has that.
Mm-hmm.
You can see those cells interacting.
On the slide.
This one?
Mm-hmm. Yeah. Hold that up for a second. Yeah, this guy, right? That's showing that.
Oh yeah.
Okay, and that little arrow that says plus, that's the activation going on. They're sending signals back and forth to each other. Okay, so these guys would have been activated. This guy does what?
It's a helper cell.
Mm-hmm. Essentially helps mount a defense against that antigen. Many different things are going to happen. It can activate other CD4s, it can activate other CD8s, it can activate B cells, it can activate inflammation. Very potent cell. We didn't talk about it much because it's also crazy complicated.
So it just helps activate a lot of other things.
Correct. And this is the cell that's infected by AIDS, that non-autoimmune disease that we talked about. And then we have CD8 cells, all right, which do what?
The cytotoxic cells.
Cytotoxic. So they're going around and they're looking for cells that have a foreign antigen on them, right? The foreign antigen that activated them, they're looking for that foreign antigen on an MHC1. If they find it, they form that perforin ring onto the cell. They inject granzyme, this potent mixture of enzymes into the cell. It destroys the protein in the cell, and then it can go on and do the same thing again and again and again. Okay? Now, let's say this is activated, and it's the next day. This CD8 comes along, and it sees this cell. It's got this foreign thing that it destroys. Does it attack this cell?
You say that again?
So it's the next day, the CD8 cell is coming along, and it sees another APC that has this, the same thing that it kills, on its MHC1. Does it attack that cell?
Yeah, I remember thinking that.
Well, Gianna says no, and she's right? Why does it attack that cell? Should it attack an antigen-presenting cell? Isn't the entire job of the cell to find problems?
Yeah.
Right? So it should be displaying problems. So that's actually not a problem for it, right? So how does it know that it shouldn't attack that cell?
Why did it do it the first
Yeah,
time?
why did it do it the first time?
It didn't attack it. It just got activated by it,
Oh.
right? So, but it doesn't need to be activated again. But why does it? How does it know not to kill that cell?
It's still?
That's foreign antigen. How does it know not to kill that? It has an MHC2, right? Which identifies it as something that can have foreign stuff on it, right? So that's how. And so the antigen, the MHC2, is activating the CD4 cell, but it also identifies this as a cell that can have something. Again, we talked about the idea of an undercover cop. Buys some drugs, gets arrested with the drugs, he goes down to the station, they take him into the questioning room, he says, look, I'm an undercover cop, here's my badge number, right? This is kind of like the badge for the undercover cop, the MHC2, that it can have this stuff that's bad. Okay, let's do inflammation. So what are the classic signs of inflammation?
Redness,
Mhm.
heat,
Swelling.
pain,
Loss
loss
of
of
function.
Boom.
Where is the person? Yes, stairs.
Something is
So like, how weighted is this exam for our grade?
I can't remember. Maybe too much. I'm not sure.
Yeah.
Does anybody remember? Is it? It's in the
I'll check.
syllabus.
Cause
I think it's a lot of points.
It probably is. There's a lot of lectures here. I mean, it's probably 85.
It's three systems.
of our
in
whole
one.
grade.
No, not 85%. Like 85 points.
Oh, I was like, damn.
Okay.
I thought I had a heart attack.
All right. So a few things here. We have two different cells that are going to help us basically signal inflammation. The first are macrophages. Right. Remember, they have some receptors that look for common themes for bacteria that are coming into the body, or for tissue damage. Okay. Those things were the toll-like receptors. And if we activate a toll-like receptor, we activate cytokines. Right? And stuff starts to get attracted if we have cytokines. So the first thing that would appear would be neutrophils. The second thing that would appear, and this would only appear if there was cytokines for a long time, if we had a chronic inflammation, would be monocytes, which become macrophages when they get there. Okay? So we're starting to attract stuff to the region. Okay, and so that's going to help deal with the infection. In the meantime, we have also some cells that release histamine, which are called
The mast cells.
mast cells. They're related to basophils, but they're cells that live in the tissue. So they're basophils that live in the tissue. These cells release histamine,
Which causes hyperemia
mm-hmm.
and vasodilation.
Yeah, so it causes an increase in blood flow, right? It causes vasodilation, right? And increases capillary permeability.
And it causes swelling.
Okay. So let's think about what we're talking about here. So vasodilation, what happens to blood flow? Increases. So that's where we get the redness and the heat. Again, you can see this if you've ever been bitten by a wasp or I was going to say a butterfly, but, or a bee. You'll have the almost immediate redness, right? They're basically injecting histamine, or you often have a really quick histamine reaction. So the redness and heat occur. The capillaries start to get a little bit leaky, so liquid starts to leak out of the capillaries. That's where we get the swelling.
Oh, what's that mean?
Okay? If the capillaries are permeable, that also helps us with diapedesis, right? Which is that whole process of the neutrophils and eventually the monocytes leaving the capillary and entering the tissue. Okay? So that accounts for the part of it. The mast cells also make numerous things that are related to damaged tissue, like bradykinins. I think the one I had you know is prostaglandins.
Yeah.
Right? Damaged tissue itself also does this, right? Or we'll come in a second here. So these are the things that are detected as pain. So you have a nerve, right? It detects those as pain. Damaged tissue itself. Are also detected as pain, right? So that's where we get to the pain. Now I can't remember exactly what I included here, so let me go back a second. Yeah, I didn't do the axon
I
reflex,
was
but that's
just
fine.
going to say the salad.
Yeah, we didn't do the axon reflex. But the axon itself sends out signals that tend to promote more inflammation, right? So inflammation does spread. So that's where we get the loss of function, where the inflammation spreads over a large area over a period of a few days. All right, so that's the basics. Let's go through and make sure we didn't miss anything with inflammation, because there's a lot of stuff here. So I'm saying, we talk about the toll-like receptors and cytokines. The mast cells can, yeah, that's fine. The mast cells also have toll-like receptors. You see them there? It says pathogens and it says TLRs. So they can also detect bacteria. Right, we're pretty good there. What common drug blocks prostaglandins?
Aspirin.
Yeah.
Which is made from willow bark.
Yes. Yeah.
Why do you have that?
Because
It
it
says
was down my
that.
notes.
Yeah. That's how it was first discovered. And then once they don't make it from that anymore because the concentration's not that big. But that's where they first discovered.
What is it block?
It blocks prostaglandins, which is common pain signal. So it's just kind of a common pain signal throughout the body. So it also goes to other places besides inflammation. So again, this diapedesis occurs. The leaky capillaries make it easier for the neutrophils to leave. And so there is a whole process of this where there's some cell signaling going on. But that's what's going on in this picture, diapedesis. Again, if things are around long enough, the cytokines are around long enough, we activate more macrophages by bringing in monocytes. They enter the tissue, convert to macrophages, and start kind of eating stuff indiscriminately.
So with the acute inflammation, you said that that's when it's like forming the pus, but then how would you describe the real difference between the acute inflammation and the chronic inflammation?
Chronic inflammation is more of a low-level, long-term inflammation.
Okay.
So if you have like an arthritic joint, there's chronic inflammation.
Okay.
If you have bleeding gums all the time, right, you have some sort of gum disease, there's low levels of inflammation. If you have a plaque buildup within your arteries, then that area there's constant tissue damage. There's a low level of inflammation. So it's kind of this same process going on at a low level, but it's built around long enough that a lot of new cells have arrived, like the macrophages, to deal with it.
Okay.
So it does kind of mature. The injury matures. If it heals but doesn't heal, right? If you have an acute inflammation that turns chronic, it's going to have slightly different cytokines released. All right, there's our complement system. All right, and again, there's several different ways to activate it. We talked about the one that's innate, and that's called the alternate pathway in this image because it was the second one that was discovered. But it's actually older than the classic pathway, which is the one that's activated by antibodies. That was the first one that was discovered. Right? And only later did people realize, oh, this is actually a very old system that's activated in multiple ways. Okay, I think that might be, you guys should be able to define an antigen, right? Define an antigen.
Oh, define,
Yeah,
okay.
define. All right, remember, it did come from something that generated antibodies. That was the original definition. But now that we know more, it's anything that's going to generate immune response. So, right, because you could activate a T cell, you're not making an antibody, but you still have an antigen being activated. Okay? So, any questions before we go?
How likely are you to curb it?
Well, I don't know. No one's taken the test yet, so.
We'll see.